Hello my dear students, this is Dr. 𝗲𝗵𝗺, your biochemistry faculty. Well, in this video we are going to discuss about a very interesting topic, recombinant DNA technology. So let's start with the discussion. The recombinant DNA technology. Now, whenever we talk about recombinant, as it is clear from the word itself, we are recombining DNA. Now, the question is, what is the use of recombining DNA? What are we getting out of that if we have to recombine? Okay, so simple question is, this RDT plays a very important role in the eradication of so many diseases. Simple example I will tell you. We will start from the basic. What is DNA? We know that DNA, which stands for deoxyribonucleic acid, or it is the carrier, heredity carrier. What is a gene? Gene is the smallest segment of DNA. Right. And if we talk about how the flow of information is there in the body, we always say that this DNA is converted into, first of all, RNA. Right. And this RNA is going to translate the information in the form of proteins. And this DNA will be converted into DNA. Okay. So this is like a unidirectional flow of information. And this process we call it as transcription. This process we call as translation. Okay, and after the discovery of the AIDS virus, means retroviruses, where RNA is forming DNA, is reverse transcription, because it is opposite of transcription. Reverse transcription. Up now. The most important point here is, suppose if a person, and we know that DNA undergoes replication, this is replication. Right. Then we come over to the protein part, because ultimately what is happening is DNA is having some sequences which is converted into RNA by transcription, and that RNA is translating the information which is hidden in the form of a genetic code. So ultimately the information is being transferred in the form of polypeptides, which have obviously a N-terminal and a C-terminal like this. Okay. Up now the question is, let's say for example, if I talk about insulin. In general, what is the function of insulin? First of all, insulin is a peptide hormone. Right? Which is having 51 amino acids. And what is the function of insulin? It is an anabolic hormone. Or in a layman's language, we can say that insulin is always released during hyperglycemia. Released during hyperglycemia means when the concentration of glucose is more. And what is its function? Since it is released during hyperglycemia, it tends to decrease the level of glucose. So it will lead to hypoglycemia. Right? There are different methods by which it is doing this process. Now coming over to this, what happens is how insulin is released, because insulin is synthesized in the body, which we actually discuss in detail in physiology, that insulin is released when it is required, because we know A and C, and beech mein there is a C peptide like this, A and B, and this is C peptide. So this is released. Okay, and that is why we say whenever we have to check, there are two very important tests in diabetes which are generally actually done. One is 𝗓𝗵 test and fasting insulin. These are the two very important tests to check in a diabetic patient, which are actually giving the root cause ki how much C-peptide is being released, whether insulin is sufficient enough to act or not. Because if we talk about the transporters, these all we have discussed in our videos, so here I will be just concentrating on RDT. But yes, I just want to give you a brief exposure that what is the use of that. Okay, so now coming over to this, if any person, because we know diabetes is also of two types, 1 and 2. One is juvenile onset diabetes, the person who is unable to produce insulin, and second mein insulin resistance is there. There are so many factors which can lead to insulin resistance. So in a nutshell, what happens is, since insulin is a peptide, if we can recognize that particular sequence which is unable to express in that patient, we can replace that. And that is what becomes the basis of this recombinant DNA technology. Means we are recombining DNA. Okay, and that is why if we talk about this, there was one scientist, Frederick Sanger. This Frederick Sanger gave the structure of insulin. And he only told that it is having 51 amino acids and it is having 2 chains, alpha and beta, right? And just after, he was awarded Nobel Prize also for this, and just after his contribution, it was the first recombinant product was humulin. Humulin, which is nothing but human insulin. It is the first recombinant product. Right. So, first of all, we are going to discuss what are the steps involved in RDT. So, what we have to do is steps. The first step is isolation of the gene of interest. Isolation of the gene of interest. Obviously, we have to select that particular sequence, okay, or selection of a sequence. Now, once halki isolation and all is a very tough process, okay, I will explain each and everything one by one. So, first of all, we have to see which gene is absent in that particular individual or which gene is unable to express in that individual. So, first of all, we have to isolate the gene of interest or selection of a particular sequence. Now, once we have selected the sequence also, now the thing is that whether it is having an origin of replication or not, because always keep one thing in mind, whenever DNA has to undergo replication or transcription, the first step is always selection of origin of replication. Whether we have to, it is like, whether 6 to 7 kilo base pairs are required, whether more than that are required, so many things are there. And isolation also, it's a very tedious process, okay. So, just to have an origin of replication, we require something to combine with DNA, because we have put on so much of labor to isolate DNA. We require that whenever we are forming a clone of that, so multiple copies should be there. So, we have to search for a suitable vector, okay. So, second step is suitable vector, right, so that it has an origin of replication. Right. So, first of all, what we will do is, we will simply isolate a gene of interest or selection of a particular sequence, then suitable vector so that it has an origin of replication. Replication mil gaya, DNA sequence mil gaya, then we form a chimeric DNA. So formation, third step is formation of a recombinant DNA is there. We can call it as a recombinant DNA or a chimeric DNA is there. Now, once recombinant chimeric DNA is produced, then we have to insert it into the host cells. That is also very important. So, insertion of chimeric DNA into the host cells for multiplication. Now, the fifth one is, now we have what? The clones will be there, multiplication will be there, and there will be a genomic library which will be constructed in the host. Okay. So, fourth step is insertion of chimeric DNA into the host cells for multiplication. Then the fifth step would be formation or construction of a genomic library. Now, the question is that this genomic library, it is possible that some recombinant clones will be there, but there is a possibility of having non-recombinant clones also. Now, if we have put on so much of efforts, so obviously we will go for those which are having recombinant clones. So, sixth would be screening of recombinant clones. Now, the thing is that, so these are the steps of RDT. First is we have to isolate the gene of the interest or selection of a particular sequence. Second is we need to have a suitable vector so that it has an origin of replication. Right? Just say for example, suppose if say this is DNA and say this is the sequence which we require, right? And here we take the vector, this vector which is having origin of replication. Now the question is how to choose the vector, okay, and how to cut DNA. So choosing of an vector and cutting of this gene or DNA requires a set of enzymes which are known as restriction endonucleases, or they are also known as the molecular scissors. And always remember, those molecular scissors are specific for a particular site. Okay, so I can simply say that, which we are going to discuss. So what happens is this particular sequence is cut and it combines with, here we can say that it is also cut like this. So this has to fit here, and what happens is we get this type of product. So what is this? This becomes a chimeric DNA like this. This plus this like this. This becomes a chimeric or a recombinant DNA. And this is done by DNA ligase, which we know is the glue of DNA. Now, this will then enter into the host cell. Like this, say this is the host cell, and it is going to multiply here. So we have to see so many copies will be produced like this. Like this, or you can say like this. So we have to search for this becomes a genomic library, or from this genomic library we search the clones or the recombinant clones where our gene of interest is located. Gene of interest is located, is present. Okay, so this is the entire set of procedure, right? Now coming over to this, first of all, how DNA is cut, that is very important. So there is a set of enzymes, as just now I told you, they are known as restriction endonucleases, right, or they are also known as REs, right. So what are these REs? These are a very important, I would say, also known as molecular scissors, right. And the most important point is they cut DNA at the specific sites. They cut DNA at the specific sites. Okay. And it was, I mean, like, there is a kind of nomenclature for these REs. And what is that nomenclature? This is, suppose if I talk about, they are given a name as per their name of the discoverer, or you can say it is based on the time. Just say, for example, nomenclature is done by as the priority of discovery of the discovery. For example, the first one which was isolated was 𝗖𝗵_R1, right. So 𝗖𝗵_R1 means EC for 𝗘𝗵_𝗵𝗺 coli, okay, R1, this is the strain, and 1 is, it was the first RE to be discovered. First RE to be discovered. Okay, similarly one more is there, HIND3. HIND3. So this HIND3 is, if we talk about the name HIND, 𝗵𝗵𝗵 influenza, and ND, D is the strain, and third is that it was, I mean, like, first was 𝗖𝗵_R1, then it was the third one. Okay, so 𝗵𝗵𝗵 influenzae, right, and D is again the strain and the third number, RE to be discovered. So this is how so many restriction endonucleases are there, and the most important point which we have to remember is that they have their specific sites. Just say, for example, I'll just show you the picture. Here we can see that. This is a very good table which can show important REs which are present. See, just say, if we talk about 𝗖𝗵_R1, 𝗘𝗵_𝗵𝗺, and if we talk about the sequence, it is GAATTC, yani ki it is going to cut DNA only when DNA is having this sequence. Then 𝗵_H1, which is isolated from Bacillus amyloliquefaciens. Here you see the cutting is here. The sequence should be like this. Similarly, 𝗵𝗵𝗵 Egyptus and HIND3. 𝗵𝗵𝗵, jiska example I have given you, you can see. And everywhere you will see there is a difference in the sequence. Just suppose if I compare this, this is having GAATTC, whereas this is having AAGCTT. Now, if the sequence is different, obviously there will be a change in the sequence, because DNA which is having this sequence will be different from DNA which is having this sequence. Okay. Now, one thing if you notice here, you can see that this type of product is produced like this, similarly like this. Okay. Or we can say that DNA is forming this type of strand. Matlab cutting is like this, that a kind of sticky ends are produced. Sticky means it's like if one strand is like this, okay, like this, the other strand is like this, like this. Okay, so these are known as sticky ends. There are some REs which form these type of ends. These are known as blunt ends. Okay, so sticky ends generally are preferred over blunt ends. Why? Because in sticky ends the cohesion would be more, right? So I can say कि sticky ends are always preferred. Ends are always preferred over blunt ends. Why? Because the cohesive forces would be more. Because the cohesive forces would be more. Right? And here I will just give you the example of 𝗏𝗏 fragment. This is the significance of that 𝗏𝗏 fragment, which actually can incorporate sticky ends even if blunt ends is there. This is again very, very important, right? So I can say that sticky ends are always preferred over blunt ends because the cohesive force would be more in sticky ends, right? So this is what is कि we have to cut DNA at specific ends. अब now the question is, ये तो चलो DNA we have cut it by selecting the respective REs. Now the question is, how much DNA has to be extracted? Okay, suppose if we have to just isolate 5 to 6 kilobase pairs, suppose if we have to extract say 10 to 20 kilobase pairs, so we need to select the vector as per the DNA, because I told you that is the first step. First of all, cutting of DNA is there, right, by REs, then we have to select the vector. Now the question is, selection of vector is there. Now selection of vector simply depends on one thing, upon the size of DNA to be cloned, right? So depending upon that, there is a variation in the vectors. Let's say if we talk about this, plasmids are there, cosmids are there, phagmids are there, bacteriophages are there, so many are there. Then yeast artificial chromosome YACs are there. Right. So this is the choice, and I will just show you one picture कि we have to, first of all, we have to select one more thing कि how many kilobase pairs of DNA has to be isolated, right? So if we talk about this, I'll just give you one common example of the plasmid which is commonly used. Okay, so this is, you can see that this is again a very interesting picture. यहाँ पे you can see if we have to simply take 5 to 25 kilobase pairs, we can extract, we can use this phage. If we use 35 to 45, we can use cosmids. If we have to isolate 100 to 300 kilobase pairs, we can use plasmids. If it is 100 to 300, again bacterial artificial, or you can say 200 to 2000 kilobase pairs tak also yeast chromosomes are there, and these are the hosts from where they are extracted. And what are plasmids? If we talk about the plasmids, plasmids are simply the extra chromosomal DNA molecules. Extra chromosomal DNA molecules which have origin of replication. This is important. Which have origin of replication in them. Very important. Okay. Now there are two examples of plasmids. One example is PBr322. Then there is 𝗵19. So P stands for plasmid, BR stands for the name of the discoverer, okay, and 322 is just the strain, and depending upon how many base pairs it can have. So PBR mein P is for plasmid. So PBr322 and 𝗵19, these are the two very important plasmids. PBR, P stands for plasmid, BR stands for the discoverer Bolivar and Rodrigues. So Bolivar and Rodrigues were the two scientists who and this plasmid is having roughly 3, 4, 6, 7 base pairs, right? And what is the characteristic feature of this plasmid? I'll just show you the picture. This plasmid has two genes, okay. If you see it clearly, it is having a one ampicillin resistant gene and one tetracycline resistant gene, okay. This is the tetracycline resistant gene, and this is ampicillin resistant. And the most important point here is this becomes the origin of replication. Now just by looking at the picture, and these are the cleavage sites and these are the REs. EcoR1 is going to cut here, HIN3 is going to cut here, 𝗵H1 is going to cut here depending upon again the specific sites. Similarly, PST is going to cut here. Now the question is, fine, DNA will form DNA will ligate with it and it is going to form a chimeric DNA. Now one example I am quoting, suppose if the known sequence of DNA goes and mixes with tetracycline resistant gene, what will happen? Obviously, that means that tetracycline resistant gene has gone because it has been replaced by DNA. So whenever we are doing that antibiotic sensitivity test, the colonies will be sensitive to tetracycline because that gene has already been taken over by DNA. On the contrary, if DNA fixes itself in tetracycline, के बाद ampicillin, so ampicillin it is allowed to, but this becomes the test how to differentiate ki where the recombinant clones have gone. So if it goes and binds with ampicillin resistant gene, so ampicillin resistant wala factor goes off. So whenever it is grown in a culture, okay, and whenever we have to just calculate that zone of hydrolysis and all, we will see that the colonies will be ampicillin sensitive. So this is what is the use of the plasmids. And this PBr322 was the first plasmid which was discovered by Bolivar and Rodrigues. So it is roughly having 3, 4, 6, 7 base pairs. And if we talk about 𝗵19, 𝗵19 was another plasmid where P stands for again plasmid. And this UC stands for University of California. And it is having some 2, 4, 6, 8 base pairs, right? So what we do is we are taking the plasmid or the cosmid or the phasmid depending upon how much amount of DNA has to be incorporated, and they are fixed. And when they are fixed, you can say that plasmid plus DNA of interest. They are ligated by DNA ligase. And what we get is, we get a chimeric DNA. Okay, now this chimeric DNA has to now enter into the host cells. Now, what is the process, what are the different processes which can make its entry into the host cells? So, the name of the processes are, it can be transformation, it can be conjugation. If it is in the form of a 𝗏, because 𝗏 also are used, so 𝗏 is there, so that is known as transfection. That also we know. ठीक है, transfection. And just write when a 𝗏 is used. Okay, these are the different processes, and sometimes electroporation is done. Different processes are done. Then host cells में they are allowed to multiply, whether it is transformation, whether it is conjugation, whether it is transduction, different processes will take place, and then we have to do the screening. And this screening is done by different microbiological tests. It can be done by PCR, it can be done by hybridization. So many are there. It can be done by blotting techniques. That is a different process. Okay, so this is what is the outlay of recombinant DNA technology. Very, very important. Now, another important feature after this is, what are the different enzymes which are used in the entire process? So, what are the different enzymes? If we talk about this, this is the history of the enzymes. Now, see, these are all the enzymes which are used. So, if we talk about, first of all, we will take up DNA. First we will take this alkaline phosphatase, right? What is the function of alkaline phosphatase? Means it is removing. So, it removes phosphate group from 5' end of double-stranded DNA. 𝗏𝗵 nuclease for the progressive shortening of DNA, because again it is very laborious to you can say take a larger DNA molecule than a shorter one. DNA ligase, which is linking, which is sealing by forming phosphodiester linkages between DNA fragments. We know phosphodiester linkage is there. DNA polymerase 1. It synthesizes DNA complementary to a DNA template. These are all the list of the enzymes which are used in recombinant DNA technology. Then exonucleases 3. DNase 1 produces single-stranded nicks in DNA. Exonuclease 3 removes the nucleotides from 3' end. Then exonuclease from 5' end. Polynucleotide kinase, which is kinase means which is transferring the phosphate group from ATP to 5' end. And the most important one are the restriction enzymes. Very, very important. Restriction enzymes cut double-stranded DNA with a specific recognition site. Right. And then reverse transcriptase, we know that it synthesizes DNA, and this DNA is also known as complementary DNA. Then RNase cleaves and digests RNA and not DNA. Then comes RNase H. It cleaves and digests the RNA strand of RNA DNA hitch. These are the enzymes. TARC DNA polymerase, it is used in PCR. PCR, I told you, it is again a very important, I would say, technique, polymerase chain reaction, just to amplify the amplicons wherever there is, because to check the disputed parentage, to check for aneuploidy, these are the techniques which are carried. S1 nuclease degrades single-stranded RNA, and terminal transferase. This is also very important. Terminal transferase adds nucleotides to 3' end of DNA and RNA used in homopolymer tailing. Because I told you, it introduces basically, sticky ends are there and blunt ends are there. Generally, sticky ends are preferred. Always keep this thing in mind. Okay. Why sticky ends are preferred? Because sticky ends give us a maximum cohesion, so that whenever DNA is ligated with a plasmid, it gives maximum results. Okay. So these are what? These are the basic features of recombinant DNA technology. Okay. Isolation of the gene of interest, right? Isolation of an origin of replication. Both are cut by the molecular scissors, which are known as the restriction endonucleases, or the REs, which always cut at the specific sites. Then they are ligated to form a recombinant DNA or a chimeric DNA. And this chimeric DNA is then by different techniques added to the host cells, where it will multiply. After multiplication, there will be a library. And that library will be full of clones, it will be full of other things which are not getting that respective DNA. So it would be like clones and non-clones. So from that we have to separate only the clones. And for that there are so many techniques which are there. Blotting techniques are there, PCR is there, hybridization is there, and we are going to get the specific clones. And then the last step should be, the last would be कि selection हो गया, सब कुछ हो गया, then expression of the clone to have a desired result, to have a desired product, because isolation and all requires so much of efforts. So everything is done in a very well planned manner, and that is what is the use of clinofragment also here. Because clinofragment are the one which is having an extra activity that what DNA polymerase 1 is having. Because there is a smaller fragment, there is a larger fragment, so it will cut and it will replace that. So these are the basic principles of recombinant DNA technology, right? In the next video, we are going to discuss the different techniques which are involved with this. And we are going to discuss about different blotting techniques, hybridization techniques, and PCR. So that's all in this video. It is a very important video and it is the basic video on RDT. I hope you have understood this video. Thank you so much.